We have sections 3.3 to 3.4, which is meiosis 1 and 2, the stages of meiosis, and also about random fertilization. So a quick overview of meiosis. In the beginning, it's pretty much the same as mitosis in regards to duplication of chromosomes. In both meiosis and mitosis, chromosomes are duplicated in an interphase. But however, unlike mitosis, in meiosis, it's followed by 2 consecutive cell divisions. Mitosis only has 1 cell division. Meiosis results in ultimate 4 daughter cells. Mitosis resulted in 2 daughter cells. Each of these resulting daughter cells has only half as many chromosomes as the parent cell, also known as a haploid. Meiosis is used to produce gametes, sex cells. So meiosis 1, overall, the first step of meiosis is to separate homologous chromosomes. And this is made up of 4 stages: prophase 1, metaphase 1, anaphase 1, and then telophase 1 with cytokinesis going on at the same time. Okay, to start off, in interphase 1, we know that the nuclear envelope starts to disfragment, centrioles start to duplicate, and then the chromosomes duplicate themselves. In prophase 1, we have the centrosomes start to move to each side. The spindle is formated, which is microtubules, and the nuclear envelope breaks down. During early prophase 1, we have crossing over occurring, which occurs at chiasmatas, which is where the X forms between the 2 homologous chromosomes. During this crossing over is where random fertilization occurs. And then later in prophase 1, the microtubules from one pole will attach to the 2 kinetochores, which are the protein structure on chromatids. Then the homologous pairs will then start to move towards the metaphase plate. So metaphase 1, the homologous chromosomes are lined up on the metaphase plate, which is actually an imaginary line, it's not a physical thing. Each kinetochore is facing one pole of the cell. In anaphase 1, the protein bonds that produce cohesion between homologous chromosomes breaks, which then causes the microtubules to pull on each homologous chromosome and separated them into separate spheres. And then in telophase, finally, each half of the cell has a complete set of haploid set of chromosomes. Now also during this step, cytokinesis occurs, in which cytoplasm is split. In animal cells, it forms a cleavage furrow, and in plant cells, it forms a cell wall. And so ultimately, the outcome of meiosis 1 is 2 haploid cells are formed. Each of these chromosomes, though, still has 2 sister chromatids attached to one another. So meiosis 2 is ultimately going to split those 2 sister chromatids.
All right, well, so meiosis 2, there is no stopping point in between meiosis 1 or 2, it just continues. And this stage also resembles mitosis. And yeah, and here the chromosomes are divided, and which produces 4 haploid daughter cells, each having half the set of chromosomes as the parent cell. And each of these cells has genetic variability, which I'll explain later when I'm going through the steps. Okay, so for prophase
Question.
Sophia.
So at the end of meiosis 1, are they haploid or are they diploid?
They are haploid.
They are haploid, so then when they're going to divide again, aren't they though?
Yeah.
So then what are they done? Do they stay haploid?
Diploid is when
They stay haploid, yes.
They stay haploid because right here we have 4 different chromosomes,
Excellent.
but then each of these is going to split, and ultimately we're going to have 4 different cells, each with 1 chromatid in it, which is their own chromosome.
Wait,
Very
there's
nicely
half of our
explained.
chromosomes.
From the start.
Gotcha.
Okay, so in prophase 2, the first step in meiosis 2, the spindle fibers begin to form with the centromeres as the 2 cells begin to separate. And in metaphase 2, you can just go to the picture. The chromosomes begin to move towards the metaphase plate, just like in mitosis, and the kinetochores are attached to each separate pole in these cells. And in anaphase 2, the sister chromatids begin to separate as the proteins holding them together begin to break down, and the chromatids move to the separate poles of each cell. And in telophase 2, the haploid daughter cells begin to form right here, and the nuclei begin to form in each daughter cell as decondensing of the chromatids and cytokinesis occurs. And each of these are genetically different from one another, as crossing over occurred in meiosis 1, so each of these has a different set of genes than the parent cells. And then just to review over, so we have prophase 2, which is where the spindle begins to set up. Metaphase 2, where the kinetochores are attached to each pole. Anaphase 2, where the chromosomes split into chromatids. And then telophase 2, where 4 daughter cells are created, which in turn produce the gametes.
So like we already went over, the meiosis will reduce the number of chromosome sets from 2 to 1. And comparing meiosis and mitosis, meiosis produces cells that are genetically different from the parent and each other, while mitosis produces daughter cells that are identical to the parent cell. Going over some of the differences between meiosis and mitosis, there's going to be like a chart that I can show you guys after this. So, synapsis and crossing over. During prophase 1, duplicated homologs pair up when crossing over occurs, and this usually doesn't happen in prophase of mitosis. In metaphase 1 of meiosis, chromosomes are positioned at pairs at the metaphase plate rather than individual chromosomes like in mitosis. And then in anaphase 1 of meiosis, the duplicated chromosomes of each pair move towards the poles, but the sister chromatids remain attached. And in anaphase of mitosis, the sister chromatids separate. So you can see in this chart, there's comparison of mitosis and meiosis. You can see that the differences at each phase.
So now the question comes up, how do we account for genetic variation? Once these differences arise, reshuffling of the alleles, which are a different version of genes that come from mutations, during sexual reproduction, produces the variation that results in each member of a sexually reproducing population having a unique combination of traits. So put simply, the behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arises in each generation. And the 3 mechanisms that contribute to genetic variation are independent assortment of chromosomes, crossing over, and random fertilization. In metaphase of meiosis 1, the homologous pairs, each consisting of maternal and 1 paternal chromosome, referring to paternal from the father, maternal from the mother. They are situated at the metaphase plate, as you can see here. Each pair may orient with its maternal or paternal homolog closer to a given pole. So that means there is a 50% chance the daughter cell will have the maternal chromosome at one end and 50% chance that it have the paternal chromosome. Each pair of homologous chromosomes is positioned independently of the other pairs at metaphase 1. So meiotic division results in each pair sorting its homologs into daughter cells independently of every other pair, hence independent assortment. To show the number of combinations possible for a daughter cell, you can use the expression 2 to the n, where n equals the haploid number of the organism, and in the case of humans, n equals 23. So does anyone want to pull out their calculator and see how many combinations are actually possible? Doing 2 to the 23rd.
There you go, Tony.
Whoever gets it, just shout it out.
8,388,608.
Right, so each gamete you produce contains one of roughly 8.4 million possible combinations of chromosomes, which is why we have so much genetic variation today. Yes?
So does that mean that, like, if I was to have, like, 2 kids, the chance of them being genetically identical is 1 in, like, 8.4 million?
That's ask for only one gamete.
Yeah, I'll get to that.
Oh, jeez.
Yeah,
Okay.
there's a lot.
Just wondering. Wait, but identical twins are the exact same because they come from the same gamete, right?
Yes.
Then how does that happen?
there's
How
a
do you have
male.
like two people?
There's also a male gamete.
Oh, I know, but like, how does that happen? Like, how did
So I'll get into detail about
no, I'm
it.
talking about identical twins. Like,
We'll
how
see.
can you get two people from like one egg cell and like?
It's split.
I didn't give him a chance to talk about
Okay,
it.
never mind.
Excuse me.
Okay.
No, Jacqueline, you're asking an awesome question, but I wanna let them,
Yeah, I was just wondering. Okay. I see.
Alright, so the picture I just showed you suggests that each chromosome in a gamete is exclusively maternal or paternal, which is not entirely the case because crossing over produces recombinant chromosomes, which are individual chromosomes that carry genes, DNA derived from two different parents. So this crossing over begins early in prophase 1 as the homologous chromosomes pair loosely along their links. And the DNA of two sister non-chromatids, one maternal and one paternal, the red and the blue, is broken by specific proteins at precisely corresponding points, and the two segments beyond the crossover point are each joined to the other chromatid. And as you can see here, by the combination of the red and the blue, there is a piece of maternal chromatid joined to a paternal chromatid, and vice versa. And this crossing over produces chromosomes with a combination of maternal and paternal alleles.
Right, as Albert was saying, each female and male gamete has roughly 8.4 million possible chromosome combinations, and that's by independent assortment. But then the fusion of the male and female gamete gives a zygote, which has around 70 trillion diploid combinations, which is why no one person in this world is the same. And that's brought by crossing over, as Albert talked about. Evolutionary significance, those best suited to the environment yield the most offspring because they're able to adapt and survive better. Then as the environment changes, population survives if each generation copes with the changing environment, and that's brought by different allele combinations that work better than the previous ones. Most differences are brought by mutations, sexual reproduction, which is the crossing over and such. And then this was first figured out by Charles Darwin, who was the first to suggest the theory of evolution.
Okay, let's do a review game. Meiosis results in how many daughter cells? Jackie.
4.
Good. Fill in the blank. Chromosomes line up at the blank during meiosis 2.
Metaphase plate?
Meiosis produces cells that are genetically blank than the parent cell and each other.
Different?
Yes. Last one, name the 3 mechanisms that contribute to genetic variation arising from sexual reproduction. Clark.
Independent assortment of chromosomes crossing over and random fertilization.
You go.